If you ask a large language model today to analyze a massive million-cell tumor data set, it's probably going to write you this gorgeous, syntactically perfect Python script.
Oh, absolutely. It'll look incredibly convincing.
Right, but then it'll silently destroy your data because it hallucinates tools that don't even exist, or it feeds the data into a real tool backwards, and the system just confidently spits out biological nonsense.
Yeah, it's a huge problem. We've been treating these models like they're, magical biological oracles.
Exactly. Welcome to today's deep dive. We're looking at a fascinating new paper from Stanford and Westlake University that finally tackles this AI hallucination problem in biology. The system is called OmicClaw. And their mission wasn't to make the AI smarter, right? It was actually to fix the problem by taking away the AI's freedom.
It's a brilliant paradigm shift, honestly, and frankly a necessary one. Because, bare LLMs fail spectacularly at long-horizon bioinformatics. They just operate in this totally unconstrained software environment.
Because the bioinformatics ecosystem itself is just historically chaotic. You have hundreds of different packages developed by hundreds of different labs.
Right, exactly.
Tool A is written in R, Tool B is in Python. One wants a dense matrix, the other wants a sparse one, and LLM just can't navigate that friction. It guesses at the formatting and the pipeline shatters.
Which is why the prerequisite to this entire paper isn't the AI at all. It's the substrate the AI operates on, which is called OmicVerse.
Okay, let's unpack this. What exactly is OmicVerse?
So the researchers realized that before you can deploy an autonomous agent, you have to harmonize the workspace. You can't have an AI jumping between a dozen incompatible apps. OmicVerse is this unified analytical ecosystem. It brings together over 100 different methods across bulk, single-cell, and spatial omics.
That's a massive toolkit.
It is, but the critical engineering choice here was forcing all of those diverse tools to read and write from a single, highly structured data format. It's called the AND-data object.
I like to think of AND-data like a universal travel adapter.
Oh, that's a good way to put it.
Yeah, you know how it is when you travel internationally with a bag full of electronics, constantly splicing wires and chaining adapters just to make your laptop work in Europe and your phone charge in Asia.
It's a nightmare.
That exhausting data conversion is basically what bioinformaticians do all day. But by centering OmicVerse entirely on AND data, they give the AI a single adapter. Everything just plugs in perfectly.
And that structure is totally non-negotiable. It's so rigorously defined.
Right. Let's break down that mechanism for the listener, because the details here really matter. How is it structured?
So in an AND-data object, your core expression matrix is always mapped to a slot called .x.
.x, okay.
Yeah. Then your cell metadata, like patient age or disease state, that's always mapped to .obs, short for observations.
Got it.
And your gene annotations are in .var for variables. So because OmicVerse forces all 100+ analytical methods to interact with those specific slots, you just eliminate all the formatting guesswork for the LLM.
A good image memory trick for you, the listener, to visualize this, think of .x as the treasure map where the core data is buried. .obs is observing the actual patients, the metadata, and .var is the varied genes.
I love that. X marks the spot, obs for observing patients, var for genes. It's a great mnemonic. And having that rigid structure is the ultimate prerequisite for reliable AI.
So to do a quick mini recap here, let's look at the bullet points. Problem 1, fragmented bioinformatics tools cause AI pipelines to crash. Solution, OmicVerse, a unified ecosystem. And the core mechanism is that everything speaks the same AND-data language using those .x, .obs, and .var slots.
Perfectly summarized.
So question for you listening right now, think about your own digital workspace. How much time do you lose in your day just reformatting data so different programs can read it? OmicVerse basically removes that friction completely for biology.
It does. But having a unified toolkit is one thing. Giving an AI the keys to drive those tools without crashing the car is another entire...
Here's where it gets really interesting. Let's talk about the brain of this operation. They call it J.A.R.V.I.S, right?
Yes. J.A.R.V.I.S is the core engine of OmicClaw, because standard AI code generation acts like a highly sophisticated predictive text engine.
It just guesses what comes next.
Exactly. You ask for a pipeline, and it statistically guesses what the Python code should look like based on its training. J.A.R.V.I.S completely abandons that speculative approach in favor of something called registry-grounded execution.
Break down that mechanism for me. How does this registry actually constrain the model?
In the OmicVerse codebase, every single available analytical function is explicitly tagged with a Python decorator. It's called @register_function.
Right, @register function.
Right. So when J.A.R.V.I.S boots up, it doesn't just rely on its pre-trained memory of what code might look like. It dynamically scans the local registry and compiles a strict JSON schema.
And that schema tells it exactly what's available.
Exactly. It lists only the callable operations, their required arguments, the acceptable data types, and the expected outputs. It essentially builds a bounded action space.
But wait, let me push back a bit. Doesn't ChatGPT already write decent code? Why does this bounded space matter so much?
Because a bare LLM doesn't know what software you actually have installed on your computer.
Okay. It's like a fake chef writing a menu. An LLM will enthusiastically write you a recipe that calls for duck eggs and white truffles.
Yeah.
And you don't find out until you start cooking that you only have chicken eggs and button mushrooms in your kitchen.
Yes. The process fails at execution.
But J.A.R.V.I.S is like a real chef who physically checks the pantry first. Because of that @register function tag, it audits the pantry and only plans a pipeline using the exact versions of the tools it confirms are locally installed.
That is a perfect visualization of the runtime layer, but it actually goes even further than just checking available tools. J.A.R.V.I.S utilizes something called prerequisite validation.
Prerequisite validation. What does that mean in practice?
It means the AI is fully state aware. It physically inspects the current state of your AND-data object before executing a command.
Give me an example of how that works. Let's look at a specific biological mechanism.
Let's take principal component analysis, or PCA. We all know that to run a mathematically valid PCA, your single-cell data has to be scaled first.
Right, because if you don't scale it, a highly expressed housekeeping gene will completely drown out a quiet but critical transcription factor. The math executes, but the biology is compromised.
Exactly. And if you feed unscaled data into a standard AI pipeline, the underlying package will often just run it anyway. It won't throw a syntax error. It just silently returns a biologically false dimensionality reduction.
And silent failures are the absolute deadliest errors in science. You publish bad data without even knowing it?
Precisely. But J.A.R.V.I.S, through prerequisite validation, intercepts that command. Before it fires the PCA function, it checks the AND-data object for a specific metadata flag indicating that a scaled layer actually exists.
Oh, wow. And if it's missing?
If that flag is missing, J.A.R.V.I.S halts execution. It enforces the biological logic itself, alerts the user, and automatically suggests running the missing preprocessing step.
It catches the silent failure before the math even runs. That is a huge leap forward. So, time for another mini recap. Let's bullet point this execution layer. First, we have the registry. Tools are explicitly defined via the @register function tag.
Right.
Second, the system is state aware. The AI knows exactly what data you currently hold. And third, prerequisite validation. It catches missing steps, like scaling data before a PCA, before errors happen.
Spot on.
So to you listening, have you ever followed an AI's instructions perfectly, only to get a catastrophic error because the AI assumed you had a specific plugin installed that didn't exist? That's the exact frustration this solves.
It's an infuriating waste of time. But, moving past the tool logic, we really have to talk about the data logistics.
Right, because modern spatial transcriptomics datasets are not small Excel spreadsheets.
Not at all. We are talking about massive million-cell matrices. If you try to feed a dataset of that size directly into the text prompt of an LLM, it will just instantly overload and crash. The context window can't handle it.
So how does OmicClaw orchestrate million-cell data without blowing up the AI's memory limits?
This is where the engineering architecture really shines. The researchers built OmicClaw on the Model Context Protocol, or MCP. They completely divorced the reasoning engine, the LLM, from the execution environment. And to manage the massive data payload, they utilize a system of handles.
Okay, I absolutely love the handle concept. It reminds me of a coat check at a world-class art museum.
Oh, how so?
Well, imagine you walk into the museum lobby wearing this massive, heavy 50-pound winter coat. That coat represents your million-cell biological data set.
Okay, tracking.
You do not want to drag that giant coat through the delicate museum exhibits, which represents the AI's prompt context. It's way too bulky, it'll knock things over, clogs up the space. So you give the coat to the attendant, and they give you back a tiny, lightweight paper ticket. That ticket is the handle.
That's a fantastic analogy.
The handle might literally just be a tiny text string, like a data_E_42.
Exactly. The massive, gigabyte-heavy dataset stays safely parked in the local machine's memory, the coat check. The AI never actually sees or reads the millions of cell counts. It only trades those lightweight paper tickets back and forth over a communication protocol called JSON-RPC.
And JSON-RPC, just to detail that mechanism for everyone, is a highly standardized, lightweight messaging system. It's essentially a walkie-talkie. The AI uses it to tell the local server, hey, apply the batch integration tool to ticket number 42.
Precisely. The local MCP server does the heavy computational lifting using your local hardware, updates the object, and then walkie-talkies back. Task complete. The updated data is now at ticket number 43.
Which means your proprietary, unreleased patient data never actually gets sent to OpenAI or Anthropic servers. Only the handles traverse the API.
It's a massive win for data privacy and security. But, avoiding data bloat is only half the battle.
Right, because if a complex analysis takes 40 or 50 steps, the chat history itself gets incredibly long.
Exactly. Even without the raw data, the reasoning trace of an AI trying to juggle 50 sequential commands will eventually induce context bloat. The model forgets early instructions or gets confused by its own previous errors.
So how do they fix the context bloat?
By ensuring J.A.R.V.I.S doesn't operate as a single monolithic agent, it utilizes an isolated subagent architecture. Rather than passing the entire messy chat history to every single tool, J.A.R.V.I.S delegates. It spins up specialized, isolated subagents for specific roles, primarily an explore agent, a plan agent, and an execute agent.
It's like a project manager running a construction site. But the project manager doesn't hand the electrician the entire massive binder containing the plumbing permits, the concrete orders, and the landscaping blueprints. That's overwhelming context.
Right. They just hand the electrician the specific wiring diagram for the second floor. And that's exactly how this works. When you submit a complex natural language query, the Explore agent first briefly queries the ANData handle to understand the current variables. It passes a summary to the plan agent.
And what does the plan agent do?
It generates a directed acyclic graph, a DAGE of the required analytical steps. Then, the execute agent takes over, running those steps one by one. The execute agent only receives the context for the specific tool it is currently firing. When the subagent finishes, it returns a concise summary back to the main thread.
It's a beautifully elegant way to manage cognitive load. Let's do a quick recap of this architecture segment before we look at the real-world results.
Sounds good.
So, bullet 1, the MCP server provides a standardized communication protocol. Bullet 2, handles. The system uses lightweight IDs, or paper tickets, instead of transferring huge data files into the prompt. And bullet 3, subagents. It breaks complex tasks down among explore, plan, and execute roles to prevent context bloat.
If we connect this to the bigger picture, this architecture shifts the AI from being just a simple chatbot to a true operating system for biological data.
So, question for the listener. How often do you find yourself breaking down complex projects for your human colleagues, but then expecting an AI to just figure out the whole massive thing all at once? The subagent system solves that.
It really is software engineering principles applied directly to AI orchestration. But the big question is, does this rigorous engineering actually translate to better biological discovery?
Let's dive into the proof of concept and the benchmarks. Because the Stanford team didn't just build this in theory. They put it through a brutal 15-task benchmark suite, labeled T1 through T15.
They threw everything at it, from standard PBMC quality control to highly complex pancreas batch integration.
And batch integration is notoriously difficult, right? You're forcing the system to mathematically reconcile conflicting data generated from entirely different labs and sequencing platforms.
It's incredibly challenging, and the performance delta between OmicClaw, which uses the off-dot agent, and a bare LLM baseline is just staggering.
How bad did the bare LLMs do?
Well, a standard model, even a highly capable one, will often manage T1 and T2, which is basic plotting or filtering. But it completely shatters on multi-step, long-horizon workflows like RNA velocity analysis.
RNA velocity. Let's explain that mechanism for a second because it really highlights the complexity. In RNA velocity, you are mathematically comparing the ratios of unspliced, newly formed RNA against mature spliced RNA.
Right.
And you do that to predict the future developmental trajectory of the cell. It requires a highly precise pipeline where the output of step 3 must be perfectly formatted for step 4.
And a bare LLM inevitably hallucinates a parameter mid-pipeline, the chain breaks, and the model simply gives up and throws an error code to the user.
So why does OmicClaw succeed where the others fail?
Because of a brilliant mechanism they call iterative repair. Because of that subagent loop we discussed earlier, if the execute agent encounters a memory error or a missing dependency during step 4 of an RNA velocity run, it doesn't crash the session.
Wait, really? It just keeps going?
It intercepts the standard error log, reads the traceback, passes that context back to the plan agent, adjusts the parameters, and retries the step dynamically. It might lower a batch size or explicitly import a missing module on its own.
It's actively debugging its own pipeline in real time. It's literally self-healing code.
Exactly.
I mean, if you look at how human bioinformaticians work, we rarely write a flawless 50-line pipeline on the first try. We write it, run it, hit an error, read the log, tweak a parameter, and run it again. OmicClaw is fully automating that exact iterative repair cycle.
It's automating the tedious mechanics of data science. But the authors also recognize something else crucial. Biology is inherently visual. You can't just look at command line outputs, you have to actually see the clusters.
So what did they do?
Alongside the agent, they built the OmicVerse web platform. And their approach to the user interface is honestly just as technically impressive as the AI architecture.
The challenge there is rendering, right? If you try to push 1 million UMAP coordinates to a standard web browser DOM, the tab will just instantly freeze and crash.
It simply cannot handle that many individual objects. So to bypass the browser's limitations, the platform utilizes WebGL.
Okay, WebGL, how does that help?
It physically hijacks your local GPU to render the data points natively. It bypasses the standard web rendering engine entirely. But the real bottleneck is getting that massive payload from the local server to the browser in the first place.
Right, because traditionally, web apps serialize data using JSON. But JSON is entirely text-based. It requires the browser to parse every single bracket, comma, and string sequentially to understand the formatting. The parsing overhead for 1 million cells is enormous.
Which is why OmicClaw uses flat buffers for binary serialization, and that is the absolute secret sauce for their rendering speed.
Flat buffers. Break that down. How does binary serialization work?
Flat buffers allows you to serialize the data into a compressed binary format. But uniquely, the memory layout of the flat buffer in the browser perfectly matches the memory layout on the server.
Wait, so it doesn't have to translate or parse text at all?
Exactly. There is zero parsing overhead. It allows for zero copy deserialization. The GPU can read the binary data directly from memory without having to unpack or translate a single thing.
It's blazingly fast.
Right.
What's fascinating here is that they paired that high-performance visualization with a persistent Jupyter-style code editor built right into the platform.
Yes, and crucially, it is memory aware. If you ask the platform to inspect a massive matrix, it won't naively try to serialize the entire object and crash your session. It safely previews just the head of the data.
Plus, it includes an embedded live terminal shell, giving you complete system-level control right next to your interactive plots.
It really provides the accessibility of a natural language chatbot with the uncompromised power of a professional IDE.
So let's do our final mini recap here. The benchmarks show OmicClaw dominates bare LLMs on complex tasks. The core mechanism driving that is iterative repair, allowing the agent to self-correct and debug dynamically. And finally, the web platform uses WebGL and flat buffers for zero-copy, high-speed visualization.
A perfect summary.
So a question to leave you with before we wrap up. If you had a tool that reliably self-corrected its own code and caught its own silent biological errors, would you trust it to run your analyses unsupervised?
It's a big question, and it really brings us back to the core philosophy of the paper. The researchers are not arguing that an LLM possesses some emergent magical understanding of biology.
Right.
They're proving that an LLM's reasoning capabilities only become scientifically useful when they are rigidly constrained by a state-aware ecosystem. When you provide the AND data substrate, the registry-grounded execution, and the subagent architecture, the AI stops being a speculative parlor trick and becomes a rigorous scientific collaborator.
It takes the magic out of AI and replaces it with discipline. We started this deep dive talking about the expectation of simply talking to a computer and having it output perfect biological truths. OmicClaw shows us that natural language analysis is absolutely possible, but not through unconstrained generation. It requires strict boundaries.
It makes the analysis executable, fully inspectable, and most importantly for science, entirely reproducible.
Which leaves you, the listener, with a paradigm-shifting question to mull over. If AI systems like J.A.R.V.I.S can now reliably handle the technical coding, the execution, the real-time debugging, and the error checking of these massive data pipelines, what happens to the human bioinformatician?
That is the million-dollar question.
If the friction of writing syntax is removed, Are we transitioning from being the coders of scientific data to acting more like scientific directors, where our primary and most valuable job is simply knowing which biological questions are actually worth asking in the first place? It is a profound shift in what it means to practice computational biology. We highly encourage you to check out the OmicVerse GitHub to see this future of multi-omics analysis for yourself. Until next time, keep asking the right questions.